So today's topic is the acute inflammation. It is one of the most important topic in the NEET MTS as well as rguhs. So we will study it through a simple story, and let me tell you this. Acute refers to quick. Inflammation refers to any response of the body to the injury. So the story goes like this. It's the body's emergency war. So acute inflammation is the body's emergency war where a cycle of events take place. So that cycle we'll study now. The, Whenever there is injury to the body, the vascular events take place. In the vascular events, the mast cells release histamine, and the histamine helps in the dilation of the blood vessels and causes vasodilation. Whenever there is vasodilation, there is increased permeability. Due to increased permeability, there is fluid leakage, and whenever there is a fluid leakage, the neutrophils arrive at the site of the injury and start the cellular events. So this is how the cycle goes. Now we'll study about the characters of the story. These are the superheroes of the story. So you have to remember this. The first one being the histamine. Histamine is released by the mast cells. It helps in the vasodilation. So next is the leukotrienes. Leukotrienes such as IL-3, IL-6, and next is C5a. It is called as the Google map of the inflammation as it leads the neutrophils to the site of the injury. So the C5a helps in the chemotaxis, helps in the neutrophil activation, and also in the mast cell activation. Next one is selectins. Selectins help in the slow rolling of the neutrophils in the cellular events. Then integrins. The integrins help in the firm adhesion of the neutrophil to the endothelial walls. And next is the PCAM1. So the PCAM1 is platelet endothelial cell adhesion molecule. It's the gatekeeper at the blood vessels. So it is present on the endothelial cells as well as the neutrophils. So this is the simple story about acute inflammation, and now we will dig into it. So let's dig into the topic acute inflammation. Starting with the definition, it is the rapid short-lived response of the body to any kind of injury. Next, the primary cell type that is included in acute inflammation are neutrophils. The hallmark event that takes place in acute inflammation is the increased vascular permeability. This increased vascular permeability lets the neutrophils to come out of the blood vessels and reach the site of injury. And next, now we'll move on to the 5 cardinal signs of the local inflammation. And these are rubor, calor, tumor, dolor, and functional lisa. Rubor, that is redness. Calor refers to heat. Tumor refers to swelling. Dolor refers to pain. Functional lisa is the loss of function. So the rubor, that is redness, is caused by the vasodilation. Calor, heat, is due to the increased blood flow, and the swelling, that is due to the vascular permeability or the edema. Dolor, that is pain, it is caused by the chemical mediators. Functional lisa, the loss of function, it is caused by the tissue damage. So these are the 5 cardinal signs of the local inflammation. Now we will move on to the 2 phases of the acute inflammation. They are vascular phase and the cellular phase. The vascular phase recruits the components of the blood to the site of the injury, whereas the cellular phase recruits the WBCs from blood vessels to the site of injury. Now we will study the vascular phase. So the vascular phase includes vasodilation and the endothelial gap formation. The vasodilation is caused by the histamine released by the mast cells. And next is the endothelial gaps. And these gaps are formed due to immediate and transient effects and reversible damage and irreversible damage. And due to this, the cellular phase now starts. Next, we'll move on to the cellular phase. The cellular phase includes margination, rolling, adhesion, diapedesis, and chemotaxis. So, this is the diagram of the blood vessel. So, through this diagram, we will study all these one by one. So, here you can see due to the inflammation, due to the acute inflammation, there is vasodilation which was caused by the vascular phase, and due to the vasodilation there is a turbulent flow in the blood vessel, and due to this turbulent flow the neutrophils tend to move to the margins of the endothelial cell, and this is called as the margination. Next is rolling. In this, the neutrophils roll slowly along the endothelial walls. This is due to the selectins. Next is adhesion. Here, the neutrophils firmly get adhered to the endothelial walls. This is due to the integrins. Next comes the diapedesis. Diapedesis is the squeezing and coming out of the neutrophils out of the blood vessel. So these are margination, rolling, addition, diapedesis. So let's study the cellular phase in detail. Now, margination is already over and we should study about the rolling. Here, as you can see, this is the endothelial cell and this is the neutrophil. And the receptor that is present on the neutrophil is CLL-Lewis X receptor. And the receptor that is present on the endothelial cell is selectin. It can be E-selectin or P-selectin. And the bond that is formed between the neutrophil and the endothelial cell is a weak bond. Now let's move on to the addition. Here, as you can see, the receptor that is present on the neutrophil is the integrin and the receptor that is present on the endothelial cell is CAM. CAM is cellular addition molecule. As you can see here, the bond that is formed between the neutrophil and the endothelial cell, it is a strong covalent bond and we can say that it has a strength like a Fevicol. Diapedesis is a two-step process. Here we see the PCAM1 and the non-specific collagenase. So these are the PCAM1 receptors. The neutrophils hold on to it and squeeze out of the blood vessel. So this helps in squeezing out and coming out of the blood vessel. And as you can see, beside the endothelial cell, there is a thick basement membrane and there is non-specific collagenase present here. And this non-specific collagenase helps in the dissolution of the basement membrane and helps the neutrophil to come out of the blood vessel. So now we will move on to the chemotaxis. The chemotaxis is the unidirectional guide to the neutrophils towards the bacteria. So this is the neutrophil. And here, the hallmark event that takes place is the actin polymerization. And another important things what we notice here is the endogenous chemoattractants and the exogenous chemoattractants. So the chemoattractants are nothing but the perfumes. The endogenous chemoattractants are the perfumes possessed by the body and the exogenous chemoattractants are the perfumes of the bacteria, such as the bacterial products. It may be lipopolysaccharides, flagellar antigen, or any such thing. The endogenous chemoattractants are interleukin 8, C5a, leukotriene B4. So these are the endogenous chemoattractants and exogenous chemoattractants. And there's one more thing happening here. Here, the G-actin is converting into filamentous actin and the neutrophil slowly converts into pseudopodia and moves ahead. Again, it converts into the pseudopodia and moves ahead. Like this, it reaches towards the bacteria. So now, we will study about the phagocytosis and the bacterial killing. So here there are three steps in the phagocytosis and the bacterial killing. The first one being the recognition and engulfment. Second one is phagosome formation. Third one is fusion. So here, as you can see, this is the neutrophil and this is the toxic bacteria. And here, the next one is, this is the neutrophil and here this is the toxic bacteria which is inside the neutrophil now. This is called as the phagosome. Next one is, here as you can see in this diagram, the neutrophil has lysosome as well as phagolysosome. So this is the fusion step. So this is all about the phagocytosis and the bacterial killing. So talking about the outcomes of the acute inflammation, in 24 hours, if it gets resolved, then it's by the macrophages. Then if it's not, then the chronicity will develop by the standard macrophages. So this was all about the acute inflammation.